more of our interviews with doctors throughout the globe in our Vasculitis Foundation project, these different grant recipients that the Vasculitis Foundation has been giving out. And we talked to another one of our doctors in the midst of a research grant right now. He's Dr. Peter Heeringa from the Netherlands. He works on the Vasculitis Foundation board in that country, raising awareness about the different types of vasculitis. So, Dr. Heeringa, thank you so much for joining us today. I understand you are in the midst of this current study. So first off, just tell us a bit about what you've currently been working on.
Okay, so thank you very much. So our studies focus on diseases known as ANCA-associated small vessel vasculitis. And ANCA-associated small vessel vasculitis are very severe diseases that are the result of an attack by one's own immune system and therefore are considered autoimmune diseases. Now, the disease is generally believed to be caused by circulating proteins, so-called autoantibodies, that are produced by cells of the immune system. And these autoantibodies are termed anti-neutrophil cytoplasmic autoantibodies, or ANCA in short. And what we know from previous work is that these autoantibodies target and activate white blood cells, and that this results in inflammation of small blood vessels in organs throughout the body, such as the kidneys, the lungs, and the skin. So specifically in this project, our aim is to study specific biochemical features of the autoantibodies that cause the inflammation. And more specifically, what we know from experimental work in other diseases, such as rheumatoid arthritis, is that these antibody proteins are coated with specific sugars. And these sugars are very important for the function of these antibodies. And what is also known is that the extent and composition of these sugar groups varies between antibody proteins. And this strongly influences their ability to cause disease or not. So our hypothesis in this project is that the antibodies, the autoantibodies in ANCA-associated small vessel vasculitis differ from normal antibodies with respect to the amount and composition of the sugar groups attached, and that this, in the end, determines whether these antibodies are able to cause disease or not. And to study this, we isolate the autoantibodies from patients' blood and characterize in detail, using sophisticated biochemical techniques, the extent and composition of the antibody sugar coating and compare the results with normal antibodies.
What sort of impact could this have on patients that suffer from the particular type of this ANCA vasculitis?
Right. So we believe that the relevance of this study for patients could actually be twofold. So first, we expect that if differences in sugar coating are detected between the autoantibodies and normal antibodies, then laboratory tests that are currently used to detect the antibodies and that are necessary for the diagnosis of the disease and monitoring of the disease course in patients could be refined. For example, based on the variability in sugar coating, our hope is that we will be able to distinguish between disease-causing and non-disease-causing autoantibodies. And this, in the end, could also impact how patients are treated and for how long. And I think the second relevance could be that the results of our project could also lay the foundation for therapeutic studies in the future. Such studies could aim, for instance, to interfere in the mechanisms that are responsible for the sugar coating of the antibodies, and by which we potentially could reduce or even inhibit the disease-causing properties of the autoantibodies. And we believe that such therapeutic interventions would be more specific than current treatments and are also expected to have less side effects. The Vasculitis Foundation, as you already mentioned, has generously provided us with funding for 1 year. And this allows us to perform the first experiments to test whether our hypothesis is correct. And these results of these preliminary studies will be presented at national and international scientific meetings, such as the annual meeting of the American Society of Nephrology, the ANCA and Vasculitis Workshop, and so on, but also to patient organizations, such as the Vasculitis Foundation, obviously, but also the Vasculitis Foundation in the Netherlands. Now, what we also hope and expect actually is that the results of this project will provide us the basis for more extended research proposals, for which we will seek funding at, for example, the Dutch Arthritis Foundation or the Dutch Kidney Foundation, so that we can continue our work on the biochemical properties of these autoantibodies.